Okay, I want to thank everybody for joining us for today's webinar, which is brought to you by Astrix, the title of which is Supercharge Your Chromatography Workflows with Fair Data Use Cases. Today's presentation is presented by Kelsey Boyle, who's the Director of Product Management at ZONTAL, and I will introduce her in just a second. We can go to the next slide, Kelsey. Before we get into the actual content, Astrix is obviously sponsoring today's session, so I just want to take a moment and tell you a little bit about who we are and what we do, why we're sponsoring these webinars. So we are a consulting company. We work with many different technology vendors in the space, like ZONTAL, and we've been doing this for the better part of 28 years. We bring a very unique blend of highly specialized strategic and technical services, as well as processes and technology agnostic approach, which has made us a very recognized and leading partner in helping companies in the life science space transform their organizations and shepherd them through what we like to call this digital transformation. And a couple of things we like to point out is of the many employees that exist at Astrix across the globe, 90% of which hold advanced science degrees. So we are very much a science-based companies ourselves, helping other science-based companies. And again, we've been doing so for the better part of 28 years. So that's just a little brief introduction into who we are. I'd like to introduce our speaker now, so we can go to the next slide. So today we're going to be presenting is Kelsey Boyle. She's the Director of Product Management at ZONTAL, and she is a chemist by training with focus on chromatography and innovative product development. And she's the Director of Product Management right now, where she helps clients with three key areas: data integration, data collection, and storage and data reuse. So Kelsey, I'll let you kind of pick up from here. I'll come back on when we get to the end of the presentation, help out with any questions, and I'm going to sit back and enjoy the presentation with the rest of our attendees.
Thank you for the wonderful introduction, Kevin. And thank you to Astrix. We've really enjoyed partnering with you and helping bring our data use cases to many of our clients throughout the industry space. For today's presentation, our agenda is as follows. We're going to open it with what is ZONTAL, and from there we will move into covering a couple of key use cases: digital methods, uploading methods to ZONTAL and sending directly to instruments. We'll follow with intellectual protection, archiving data backups and symbiosis with other use cases, such as bringing in data from CROs. Following with mergers and acquisitions. How do we unlock the insights of siloed data during the M&A operational hurdle? And we'll wrap up with research and development. With a fair data format, it's now possible to compare and visualize chromatography results from different chromatography vendors. Following the discussion of those use cases, I'll do the really exciting part. I'll jump into ZONTAL and show you a demo of the research and development workflow. Then we'll shift back and Kevin will join us again for some Q&A time. Let's jump right in. I'd like to ground us by beginning with three use cases across data management. We can kind of bucket these into three logical areas. The first one being data collection and storage, whether that be for archival, decommissioning, or preservation. The second one being data integration. When we think about data across life science, these can be found in many different places, such as electronic lab notebooks, which according to a recent NIH publication, there are over 172. Laboratory informatics information systems, as well as workflow execution. And our final bucket is data reuse, be it data visualization, secondary analysis, or the all too familiar phrases now of AI and machine learning. Life science faces its own unique challenges. When we think about the size of data and the individual data size from experiments from the early 2000s to now, they're continuing to get more complex as our techniques evolve throughout the analytical chemistry space. When we think about metabolomics or computational biochemistry, perhaps even one day quantum. How does this impact the data that our teams are tasked to then logically find ways to use throughout these three different types of use cases? Our amount of analysis has increased, and often our data itself has gotten more siloed and complex due to organizational changes as well as organizational mergers. Our velocity has also changed. as more routine analysis is required, as regulatory standards are constantly changing, as well as we exist in a global regulatory environment. This gets more complex as we have products active in 80 different countries or submissions active in 120. We have a unique challenge in formats. A lot of our vendors use different proprietary data types. If you look at mass spec, there's over 15 different proprietary file types by each of the vendors. We utilize a wide variety of techniques throughout our labs in the pharma and biopharma space. And we have unique instrument classes, as well as new instruments and new technologies coming on at any time. How do we approach challenges to soft nanoparticles as we begin to characterize this brand new novel therapy? We also are faced with retention, global data policies, which sometimes result in multi-decade data requirements that we need to retain that data for legal or regulatory purposes. And at the bottom line of everything, integrity. Us as scientists, we value accuracy and consistency, and we want one-to-one capture of everything we do. And that's what holds us as a grounding principle to how we approach our use cases. Our clients come to us with these different data use cases, and they ask us, is there a way to solve this problem? And what we find is a lot of our clients are currently solving these problems individually. They have individual use case and individual personas that all access different internal tools or utilize different ways to achieve those grounding things. And today there's a different way to accomplish this. It's through bringing in a digital platform, ZONTAL. If we think about one place, one source of truth, where we can take in all the information from all those different data producers, all of those experiments, where we store our data itself, and have a launch pad for automation and analysis, as well as workflow execution. That's what ZONTAL is. And how does this actually work in the real world? We have instruments. Today, we're going to talk about chromatography instruments, but we take instruments from all over labs, and not just from the pharmaceutical industry, from other industries as well. So we can take those instruments, whether they're on your manufacturing floor or whether they're in your laboratory. If they produce data, we can bring it in. We can automate this process. We can create data watchers. And then we want to create a fair data platform. Fair data has been around for a while, was first brought and published in 2016. And we know the acronym stands for findable, accessible, interoperable, and reusable. And really it boils down to making data searchable in a way that other people can use it and find it. So we've partnered with the Allotrope Foundation to align under their standard models and ontology, and we're heavily involved in those working groups to ensure a place and mesh us in how fair data is going to move forward today. Through our process of pipelines and integrations, which we have over 400 different integrations today at ZONTAL, we're able to take and enrich with metadata, whether that be descriptive metadata, administrative, and then take in that and bring it into a searchable platform, which is ZONTAL. After that first step, the system is really providing an enterprise data archive. And when we think about this, there's really a difference here between preservation and archival. Preservation means continual access and a pathway to an organizational change. And ZONTAL is able to accomplish both. We are archiving and preserving. As we move forward through our pipelines and integrations, another key thing that I like to remind clients is this is your data. You can take it from ZONTAL into other third-party platforms like Spotfire, and we have a head of analytics that is excited to talk about those opportunities with you, as well as you can utilize all the tools inside of ZONTAL itself. ZONTAL is the platform, and from there, we've developed different hubs that allow you to utilize the data inside of the platform to answer our use cases that we're going to go through today. Throughout those different hubs, you now have a solution that allows you to accomplish all of your use cases through one common platform, whether that be life science analytics, creating a digital laboratory in which we can directly send information down into instruments, or digital preservation. So let's look at these use cases. We're going to start with a chromatography workflow. Throughout chromatography, there's many different points in which data is created. There's also valuable information that we need for regulatory purposes or for R&D purposes throughout the chromatography workflow. We have our method, we have the analyst itself who's performing the request, we have data and results. Right now, we capture these in many different places, and sometimes we have legacy tools that need to be decommissioned, or we're bringing in tools from other places, and so this information is often siloed. Let's look at just one spot within the chromatography workflow to start. Let's start at the method. And the method is your grounding recipe for everything we do within chromatography. If we break this down into one problem statement inside a method, what happens when we cannot replicate a method? I personally was a grad student who worked on an Agilent 1260, and I faced challenges in my own chromatography back when I was a grad student with methods. If I couldn't get something to work, it took a lot of cumbersome root cause analysis to get something to work eventually. So today, what happens in the laboratory is if we have, for example, a new CRO that we are partnering with, we have a method and it's stored in our electronic lab notebook. We go on our instrument and we want to execute that method. What happens if our new CRO has a different vendor of equipment? What happens if they're using different versions of software? What if we compare the data and there's no match? How do we manually transfer this method and avoid any hiccups in having to approach post-approval changes on your chromatography? Imagine it differently. What if you had digital method transfer, where you could upload your method from anywhere and it's no longer siloed, it's now inside of one place, execute that method, compare the data, and match it. This is what some of our clients are accomplishing today. You might ask, how are they accomplishing it? And that's through an end-to-end process. We're able to send methods directly from an HPLC, upload it into ZONTAL, convert it into a digital method, and users can search and find a method to be used through that enhanced metadata. We can use properties such as site or analyst. We can even make it chemistry aware by searching for a SMILE. Our method can be downloaded and HPLC parameters can be normalized to minimize instrument-to-instrument variations such as dwell volume. And then our method is able to be executed in a new CDS system or project. What does this actually look like? Here's an example where two clients have taken chromatography between different softwares, one being an Agilent 1290 and one being an Acuity system. And we know between these differences in the hardware on these instruments that there is a difference in dwell volume, which we're able to observe in the chromatography viewer that is housed inside of ZONTAL. Later today, during my demonstration, I will show you around the chromatography viewer and what capabilities you have directly inside of ZONTAL without taking it back to the original third-party software. Our next use case is R&D experiment intellectual property. This is a quote from one of our clients. If it weren't for ZONTAL, we would have never known these experiments existed. We're recently beginning to have many clients reach out about intellectual patent use cases. And if you look at chromatography and patents on a quick PubChem search, there are over 31,000 different patents that contain the word chromatography. Chromatography is a vital aspect of intellectual property. So let's go back to that chromatography workflow. In first to discover legal proceedings, the data and results and proving when this instrumental work was done is vital. That work often involves chromatography. If we bring legacy data and results, as well as new data into results, into our one source of truth, we now have a place that can be utilized by different personas throughout your organization. Oftentimes, I find clients have different departments that don't always speak or unify very well together, and they have different personas who have different needs. But within ZONTAL, with access-based user controls, you can create a platform in which different users can use the same platform for different use cases. So let's look at it this way. If I'm looking for searchable intellectual property, rather than individually printing out PDFs from legacy decommissioned ELNs, if those legacy and current ELNs exist in one place that is searchable, it is a much easier exercise to hunt down something I need for a legal proceeding or regulatory use case. I can also bridge that, and I can take in information and data directly from my chromatography instruments. Think about the power of coupling this all together and making it searchable all through one click. That's exactly what our clients are utilizing ZONTAL for today. I mentioned earlier that ZONTAL is now chemistry aware. And what I mean by that is you're able to search by structures, you're able to search by smiles, and this adds in a layer of metadata that is extremely useful for those who are searching for something particular inside of the data that they already own. Here's a screenshot of what our platform looks like. And here's a list of some of the things that provide value to our users. Audit trails, data retention policies, these can all be set up to meet the needs of your global data policies. We're able to have a centralized metadata catalog, as well as we value data security and user roles. Let's look at our next use case. Mergers and acquisitions, chromatography workflow. We know that when we have a merger or acquisition, there were sometimes years or even decades worth of legacy work that were performed at that newly acquired company. How do we bring in that information and actually get something out of it? Whether that information is the methods, whether that information is the data that was produced, or whether it's the analysis or the request itself. When we're thinking about how to merge in that M&A asset, you might have a difference in the ELN that new asset is using, and it begins to be very complex when you're trying to maintain 6 different ELNs at once, and you would love a way to take that information and bring it out, and ZONTAL is the perfect place to do that. You can take out the chromatography experiments themselves through taking the legacy data. We can export that directly from the instruments, or if you have it in different storage, we're able to partner with you to come up with a solution on how to architecture that into ZONTAL. We can pair that with your current instruments and results and pair that with your current ELN to give you one source of truth, being ZONTAL. And again, that can be accessed by many different users, whether that's somebody in your competitive intelligence teams, whether that's somebody who is a regulatory user or a legal team user, or whether that is somebody within who is actually generating the science themselves. Think of how much power could be at your fingertips if you could check and see if an experiment had ever been done on a similar molecule before, whether that was legacy data that was acquired from a biotech or whether that's something that's currently being done today. And we can also see pieces such as audit trails and ELN attachments and the information from the results itself. And we can view those results directly inside of ZONTAL, as well as we can store raw data, so you could open it back within the original software that it was generated on. And inside of ZONTAL, we've also made that data fair. So we will also give you a way to export versions of it in an ASM file. Our final use case before we jump into our demo is research and development. Chromatography is a foundation of research and development. With chromatography, we do key things such as structure elucidation, quantification. We develop those critical methods that are then used in CMC for reg space, impurity analysis, reaction progress and scale up, and product characterization itself. Another search within PubChem, chromatography, your literature hits are over 782,000 different publications throughout the space for chromatography. For chromatography workflows, again, we have all those different touch points, and we can bring it all into ZONTAL. Here's an example of metadata extraction, and this one is specifically for max links. When we think about chromatography, we do cover both the initial hardware side being liquid chromatography, and then we also have different ways of handling many different types of detectors utilized throughout the chromatography workflow itself, whether that be UV-Vis DAD detectors or PDA data, as well as mass spec. And so throughout our metadata extraction, we're able to enrich and ensure that our metadata catalog aligns with your use cases. We are also able to process custom fields, as we know that within many instruments workflows, there are unique custom fields to your projects. We are continually able to partner with you on your requirements for your projects, whether that needs comes from how we organize result sets or channel sets, or whether that being what is most important to you that we parse out through our pipeline. And today, inside of ZONTAL, we also have a chromatography viewer, which I'm going to show you here in just a moment. So this is one of the most powerful tools within our application viewers. We also have a specific viewer for mass spec, as well as other techniques such as NMR. And ultimately, ZONTAL is really something where we're able to stack together different platform modules to give you those functionalities to enable each of those use cases as you see fit. If there's a project you think that we might be able to partner with you on, please reach out. I'd love to hear more about your project and how we could help you accomplish that together. All right. I'm going to jump over to our demo space now. So inside of ZONTAL, you will be greeted with a homepage landing screen, but I've pulled us into one of our workspaces, which is where we're going to start today. And what I want to start with today is taking two different vendors' chromatography results and comparing and contrasting them together. So for today's purposes, I've chosen to take Agilent results as well as Empower results. And I'm going to show you what this looks like in terms of manually uploading a Empower results file. In theory, we architecture a lot of our clients, so this is done automatically through file watchers and other different pipelines, but you can also bring results manually into ZONTAL. So I'll show you what that looks like. So we can take in proprietary file types from both Agilent and Empower. We partner directly with Astrix. They have a beautifully designed Empower connector. And so the Empower connector is able to produce a ADF JSON file, and it is also able to produce soon a direct ASM file. So this is a file generated by one of our partnerships with Astrix. They create this file off of our Empower. If I was to open this up, it is, human readable in that we've got our JSON file here. But that doesn't have as much value to me as something that would I'd be able to visualize and actually look at the chromatogram. So let's go ahead and upload this. I can add properties here if I want in a manual upload, but and then the second that I've submitted that, I can see when it was created on. I can see whether or not it's loaded into Data Hub, and I have information about this file. I have information about the size, number of files, and I can actually place in retention holds that can be programmed in for different pipelines. So since this is just a manual ingest, it's not going to have any of those tags. It takes a couple of moments, but soon we will have this file as an archive. And then I'll be able to view it inside of my chromatograph viewer. But let's take these files and compare and contrast it with an Agilent file. And within my platform, I can do a bulk operation so I can view more than one file at once. I'm going to open up my chromatography viewer. Within my chromatography viewer, I'll have an option to begin to show the data. So I can pull up the file that I've just loaded into ZONTAL, and then I can pull up other files that I already have inside my space. So now I have my two different runs here, and I can begin to overlay the graphs, as well as below, I'll have information on the individual peaks. So let's say first, I want to just overlay the graphs, look at how my peaks are aligning. Afterwards, let's say I want to do a deeper level of analysis on only one. I can take off my overlay. I can display peak labels. And this is an interactive graph here, so I'm able to zoom in, look at my resolution, decide if that'll meet my SOPs for my method development. And I can also go down to my table and see key parameters that's useful for a chromatographer, such as peak height, percent area, retention time. And all of this is also exportable. So I can download my plot as a PNG to use in presentations. So if I go back and I want to overlay these, I can create an image, and now I have a new file. This type of quick imagery could be extremely useful in your presentations when you're looking across different sites or across different instruments or across different methods. And with that, I'd like to bring us back together for some question and answer time.
Great. We're going to go ahead and get the audio recording part stopped. So let me go ahead and get that stopped.